Right, so we're going to cover enzymes today, begin today, and then finish it on our next meeting. But there's a lot of hesitation to learn this topic of enzymes. It's considered one of the more difficult topics. It's actually my favorite. It's what I did my PhD in. But I'm going to try to make it easier for you. I'm going to try to make it something you can easily understand, put it in terms you'll understand, and use a lot of examples and analogies and one rather funny story about a dog going to the prom to explain how this works. Okay, so first I want to ask you, what does an enzyme mean to you? Right, there's lots of great answers to this, but I'd just like a few of them from the audience listening. What does an enzyme mean to you? And you're probably right in your answer, and I don't mind wrong answers as well, so we can correct those misconceptions.
It speeds up a reaction.
They're catalysts for organic reactions.
Right, so it speeds up a reaction. Right, another way of saying that is it's a catalyst for a reaction. Right, they're generally organic chemistry reactions or biochemical reactions. All correct so far. What else do you know about them?
They don't get used up in the reaction.
Right. They do not get used up in the reaction, right? So catalysts are not consumed in the reaction, right? They're regenerated. That's true.
Don't they break down molecules?
They have the ending ASE, so like lactase.
Okay, they break down molecules. That's one thing enzymes can do. It's not their only ability, but that's also true. They generally have names that end in ACE. That's true as well. Right, these are great answers. You're covering half the things we're going to cover today. I'd like to get an idea of what you know before we get into it. And if there are any misconceptions, we can clear them up. So far, you guys are batting 1,000. Sounds great. Anything else?
They have specific molecules that like specific enzymes can bind to.
So they're specific about what molecules they react with. That's true. Right? Those we often call substrates. Right? Those are the name for the reactants for these reactions. That's absolutely correct. They're generally specific about that. Anything else? Right, what are they made of?
Proteins?
Proteins, that's a good answer. Is that the only answer?
No.
What else might they be made of?
Nucleic acids?
In particular, RNA, right? Not DNA. Right? So RNA, which is a nucleic acid, that's true, but it's in particular RNA. So RNA can also constitute an enzyme. So some are made entirely of protein, some are made of entirely an RNA molecule, and some are combinations of both. Right? And you've probably heard of one of these that's a big combination of both RNA and protein called the ribosome. Right? It's the enzyme responsible for assembling your proteins. Right? It's a really big complex structure made of mostly RNA, but some proteins involved as well. So it's an enzyme that's made of both of those parts. Now the vast majority of enzymes are made of just protein. That's true. Okay? But they're not only made of protein. That's the point I want to get across. And you said these are catalysts that speed up the process. That's an excellent way of saying it. And they speed it up not by a little bit. I don't mean by 1 to 2 or 10 times faster. They sometimes speed it up by hundreds to millions or hundreds of millions or even more times faster. As you see in the bottom, some of them are 10 to the 20th fold faster. That's the rate of increase, which is an incredibly large number. Right? And some are not so large. Some are only, say, 1,000,000-fold faster. But from a human point of view, that's a huge improvement, right? So these enzymes allow the reaction to take place much more frequently than it would if the enzyme was not present. And that's a point I want to make is when we say we speed up a reaction, be careful what you mean by that. Because I don't mean the enzyme makes the things move faster. Right? That is not what I mean by speed up a reaction. The chemistry step still occurs at the exact same rate. Right? The enzyme doesn't speed up the movement of molecules around. That would be temperature. Right? What the enzyme is doing is allowing this reaction to take place more frequently or more often. Right? So the same reaction happens, it just happens more often in the same unit of time. Right? And how it accomplishes that can be described by the graph on the right. So let me walk you through this graph because we're going to see lots of graphs. And the first thing you should do whenever you see any graph like this, it's an x and y axis, is you should read what's on the axes. Read what the labels on the axes are. Now, of course, if it has a title, you could read that as well, but you should always look at the axes first before you try to understand what it's showing. So what we have here on the y-axis is energy, right? And I want you to think of it as how much instability a molecule has, right? Not the word energy is kind of a misnomer. It's kind of a catch-all of things, but what does it really mean? So I want you to rewrite, instead of the word energy there, you can also think of it as the amount of instability, not stability, but instability in the molecule, how unstable it is. The higher on this graph, the more unstable the molecule is, the less likely it likes being in that conformation, if that makes sense. Right? And on the x-axis, it says reaction coordinate. This is not time. Right? This is not a linear time axis at all. What it's representing is sort of like time, but it's not time itself. It's as the reaction proceeds, what happens to the energy or the instabilities of the molecules involved. Right? So as the reaction happens, right, whether it's an enzyme or without, just as the chemistry happens, what happens to the energy of these molecules? That's all this means. So it's not necessarily a time frame or linear time frame or any scale of time that's uniform. It's just as the reaction proceeds. That's all this is saying. So the distances on the x-axis don't mean anything. It's just as the reaction proceeds, this is what happens to the trace of energy of these molecules. So let's follow along. So we can start at the beginning. We have what we call our reactants, or if there's an enzyme present, we call them substrates, but it means the same thing. Right? So in this example, we have some sugar here and some oxygen. Right? So our carbohydrate and O2, perhaps we're oxidizing this carbohydrate completely or burning it. You could think of this as a wood burning, right? Because that's mostly what wood is carbohydrates. And so as you're doing that, you can do it really, really slowly in the cells. This is our metabolic process of oxidizing sugars. And when we're done with that, we get CO2 and water, just like we would with combustion, right? Just on a much more controlled manner. But if you can imagine it happening all at once with one big enzyme, I'm generalizing here for any enzyme, is you start out with some energy on this y-axis. These are the static energies of the starting materials or the reactants or the substrates. And when we're done, we end up with some products. In this case, our example has CO2 and water, but we can have whatever the products are, and they also have a specific energy, right? They have an amount of energy in the molecule, and it's a fixed point on the y-axis. So what's happening is we go from reactants to products, right? And the two paths are shown here, one in blue and one in red. And red is what happens without the enzyme present. Right? So you see the energy here has to increase initially quite high, right? And then it goes over this hump and then goes down all the way to the products, right? We call this distance from where the reactants started to the highest point you reach the activation energy. We have to put some energy into this system in order to get the reaction going. And this is true whether you have an enzyme or not. Any chemical reaction generally works this way, right? So if you can imagine you have a piece of wood sitting there, right, or gasoline sitting there, or anything flammable just sitting there, it doesn't spontaneously turn into CO2 and water. You have to provide some energy to get it started, even in an oxygen environment. So if you can imagine gasoline or a piece of wood or kerosene or any fuel you have sitting there, you must provide a spark or some input of energy to get it started. Right? And that's what this activation energy represents. Right? It's the amount of energy required to get this reaction to start. Once you get it started here, one of two things can happen. Either it can go back to back to reactants, right? Which a lot of times it does in the enzymatic sense or in chemical reactions, or it goes over the hump and turns into products. This one is a more favorable because it falls farther, right? It's a lower energy place down here than it was falling this way. So a larger drop is more favorable. So this reaction tends to move to the right, given the initiation or activation energy. So it tends to go from a higher energy state to a lower energy state. So this is downhill overall, right? So we're here and we end up here at the products. So this is a favorable reaction, right? But in order for it to occur, it can't just simply become the products. You have to add some energy to get it started. That's our activation energy. So what's the point of an enzyme, right? What an enzyme generally does, and this is true for the vast majority of enzymes, it's not true for some of them, but for what I want you to know, in general, an enzyme will lower this requirement for an activation energy. It did not move the reactants on the y-axis. It did not move the products on the y-axis. Their inherent energies are consistent. They don't change. That's the energy of the molecule under those conditions. If I add an enzyme, all I do is change the path of getting from substrate to product, or reactant to product here. Right? So I just change the path to get there from a very difficult hill to climb, requiring a lot of input of energy from here to here, whereas with the enzyme, I only have to put in a little bit of energy, a smaller spark, or a smaller input of energy to get it over this hump. The enzyme, and this is key, I want you to remember this, does not change the energy where you start and end. It just it decreased the requirement along the path to get there. Right? It's an easier hill to climb over with the enzyme. So the activation energy in the absence of the enzyme is quite large. That's what this is showing. And the activation energy in the presence of the enzyme is much smaller. But the overall change from start to finish remained the same. Right? The start to finish is often called the delta G, if you hear it called that way, right? Or the free energy release of the reaction. That does not change. An enzyme does not change delta G. It does not change the overall energy or the overall dynamics from start to finish. All it does is change the path along the way. It makes it an easier path. So the activation energy requirement was decreased. That's what enzymes do. Right? So I'm asked that 50 different ways. Does it change the energy of the reactants or substrates and the products? And it does not. It just allows you to get from one to the other over an easier path. Right? And like you had said earlier, the enzyme is no worse for wear. It's not consumed in this process. It just provides a platform for this to happen. Right? And then the enzyme is ready to do this over and over again. And that's what we mean by an enzyme speeding up a reaction. It allows the reactants to turn into products more frequently than if it wasn't present, because this would be a much tougher hill to climb. It would occur less frequently. So when you say a reaction is sped up by an enzyme, I don't mean things are moving faster. I don't mean the chemistry happens faster. I mean the overall reaction can occur more often or more frequently. That's what we mean by that. All right, any questions on this graph so far? Because we're going to build on this.
Yeah, how is it that it doesn't make it, like I understand what you're saying, that it doesn't make it faster, it just allows it to happen more frequently. But like on the graph, isn't that just showing a few reactants going to products? So wouldn't that be like one reaction?
The graph is showing the progress of any one reaction, correct. But because that lower trace, the blue line, is easier to get over than the red line, right? It takes less of an energy input to get it started. And the lower your energy requirements, the more often some random energy from the environment, perhaps the energy of motion or heat or whatever it might be, is allowing it to get over that hump easier. And the easier it is to get over something, the more frequently it will occur.
Okay.
Right. So what you're saying is correct. It's just trying to visualize it in a way that will make it sense to you. But good question. Okay, so let's talk about the enzymes themselves. Sorry, go ahead.
I may have misheard you, but I think you said the graph without the enzyme, since it has a further drop, it's a favorable reaction. Why is that so?
So the drop I'm talking about is not necessarily from the top to bottom here. Yes, that's a very long drop, and it would rather fall here than it would fall on this side more often. But we can ignore this peak because it doesn't really matter how high this is. The true change in the reaction, whether there's an enzyme or not, is from here to here, right? From start to finish. And because we end up at a lower point than we started, then this is a favorable reaction. Not because this peak is really high or not so high, or the relative size of these, but that's sort of saying the same thing, right? But from where we started to where we ended is a downhill progression. If we started here and ended up, say, up here somewhere, that would be an unfavorable reaction. That doesn't mean it can't happen, it's just less favorable.
Okay, that makes sense. Thank you.
And another way of saying that here is imagine running this reaction backwards. Right? Some reactions are irreversible, like this one, but some are completely reversible. In fact, most of them are reversible in the presence of enzymes. So I could run this reaction. You see, it doesn't have to climb a huge hill. It's not so bad. It's just this part here. Right? So it could go backwards, not this particular example, but many reactions can go backwards, and we'll talk about that as well. Okay? So the first thing we need to discuss is, how does an enzymatic reaction happen? What are enzymes? Right? Well, we said they're mostly protein or RNA or a combination of the two, and we'll focus mostly on the protein ones here since they're most abundant. And you can say they're put together as parts, right? You've learned what proteins are. There's a string of amino acids put together and they fold up on themselves. But sometimes that's all that's required, right? Just the amino acid string or chain is put together, folds up on itself, and it forms a functional enzyme. Those are called simple enzymes, right? There's no cofactors, no additional parts needed. The activity is solely possessed by the amino acid chain itself. When that amino acid chain folds up into its tertiary, possibly quaternary structure, if it has multiple subunits, when it all comes together, it can function as an enzyme. Those are called simple enzymes, nothing added. A lot of times, the amino acids themselves don't have the required proficiency to do the reaction. They may need some help. Like they may need an iron atom bound, or they may need a biotin molecule, or a vitamin C molecule, or some other cofactor that's required. So if there's another thing involved, we call it a cofactor, right? It's like kind of like a co-pilot, right? It's required to be added to my enzyme, right? So we call what's the protein folded up without its acquired pieces it needs an apoenzyme. Apo meaning before, or not yet assembled, right? So an apoenzyme is not yet fully functional. It's the protein portion, it's folded up correctly, but it has yet to acquire its copilot. Right? So when the cofactor, sometimes called a coenzyme, when it's a very specific thing, we'll talk about those in just a second. When it acquires that, this cofactor, and all the parts it needs to function, maybe it has more than one of these, it's now called the holoenzyme. Hol for holo, right? Everything's there. So apo meaning before, it's not quite assembled. It acquired its pieces it needs. These are other molecules, not necessarily amino acids, usually some metal or some vitamin. Right? When it acquires that, it becomes the holoenzyme. It's now functional. It can bind to its reactant or substrate. Right? Those cofactors can be a lot of things. They can be just metals. Right? They can be organic functional groups, kind of like a heme group, or a biotin molecule, or vitamin C, like I said, or other B vitamins. Right? Or they can just simply be a nickel atom. Right? Or some other molecule. So anything other than amino acid chains, we're going to call cofactors. Right? And then very specifically, if this copilot we add, if the cofactor we add is of organic origin, which is what this class is about, right? It's all these organic molecules. So basically I mean everything except the metals. Right? So all those vitamins we talked about, but not the metals. Right? So if it's just a metal, we'll call it a cofactor. Right? But if it's an organic origin, such as many of these other molecules, we'll call it a coenzyme. All coenzymes are still cofactors. Right? It's a subclassification. So all coenzymes are still called cofactors, but the cofactors that are not coenzymes are the metals. So it's a very simple distinction between the two. It's either a metal ion or it's organic in origin. Right? So if it's organic in origin, our cofactors also get the label coenzyme. And like I said before, we put the apoenzyme, which is the inactive form, together with our piece that's not of protein origin, right? It's by itself can't do anything, but put together it makes the holoenzyme, which is able to function as an enzyme. That's all this means. So simple nomenclature is all we're talking about here. If it does not require a cofactor at all, right? If this were functional on its own, then there's no need to call it an apo or holoenzyme. There was nothing to assemble. It's just a simple enzyme. Right? It works all on its own. Right? And some enzymes have cofactors and some don't. I just want you to be aware of the terminology. Okay? The next thing I want to talk about is how we name enzymes. And this has changed over time. There's some very old studying of enzymes that were done hundreds to thousands of years ago, right? And they called these things by what they did, right? We still do that today, but we have a more rigorous naming system like we do with all the organic compounds you've learned how to name. So in the old system, they named it by basically what it did. So some of the earliest enzyme discoveries discovered were the digestive enzymes found in the stomach, right? And you couldn't exactly ask someone to borrow their stomach. I want to extract some enzymes and do some work with it. So they used their bodies as their own laboratories, right? So they would take a piece of meat, right? Tie it on a string, swallow it, right? Don't chew it very much, but try to swallow it. Be a small piece of meat. And then they'd let it sit in their stomach for, a minute, 10 minutes, an hour, whatever it took. And then they'd retrieve it by pulling up the string, right? You couldn't do this on a volunteer. They weren't going to volunteer. But you do it on your own stomach. That's how they did this. They would pull the piece of meat up and analyze the results. And it was clearly digested. Something was tearing this protein, which was mostly the meat they were eating, the protein apart. It must have been something in the body tearing it apart, right? And those were named pepsin because it came from the stomach. So peps means stomach, right? And trypsin because it cleaved things. And they named them all these different names. And they usually ended in "in," that "in" ending. And that's the old convention. And a lot of those enzymes have retained those names like pepsin and trypsin and chymotrypsin because they've been studied so long, the names have stuck. In the newer naming system, like you guys told me a minute ago, they tend to end in "ase." So this "ase" ending indicates an enzyme, kind of gives you a sense of action, right? So it kind of tells you this is an enzyme by this "ase" ending, with the exception that some of the really old enzyme names have stuck, like pepsin. Okay? So this modern way of naming things has this three parts. So it has an "ase" ending. It tells you the type of reaction it's doing. In this case, it's transferring something or it's dehydrogenating something, right? Remember, this was oxidation, right? And in the beginning of it, it tells you what the substrate might be. These are great enzyme names. Alcohol dehydrogenase. It's an enzyme that oxidizes alcohols. Right? Great name. So it tells you everything about it. This one down here, carboxyltransferase. It's an enzyme that transfers carboxy groups. Right? Great name. Some of them are not so great. And we'll talk about a few examples that could be improved or should have had a little more thought put into them. Right? But generally tells you what the substrate is in some manner, the type of reaction it's doing to that substrate, and then we append the ending "ase" to tell you it's an enzyme. Right? Fairly simple. That's how we named them. Some of the names are a little more cryptic, like pepsin really doesn't tell you anything about it other than it's maybe in the stomach. Right? But this more modern convention gives you more information. And that's how we'll refer to these enzymes. Will I ask you to memorize a bunch of enzyme names? No. I just want you to understand the analysis of the naming scheme. All right, we'll learn some enzymes when we get to metabolism for your last exam. All right, so these enzymes, we said, catalyze a reaction. They're not consumed in that reaction, but most of them are completely reversible. So we can have it go the forward or the reverse direction. I'm going to go back a slide and explain that. If the energy of the reactants here, or the substrates, and the energy of the products, if these 2 points are rather close to each other, right? This is a rather large drop here from here to here, right? But if this point was higher up, if these two were relatively close, then the energy difference from reactants to products or products back to reactants would be very small. And that small energy difference gives you the idea that this thing could go either way. It's not really favored very much in any one direction, right? And that's why a lot of them don't have these huge drops, and they're completely reversible. Why would it go one way or the other? It could depend on the situation in the environment of the enzyme. Maybe the pH is favoring it in one direction, the temperature, the concentrations of reactants and products, Le Chatelier's principle, if you've heard that before. All those things can affect the direction. But what we're talking about here is under ideal conditions, everything else being equal, it'll tend to go in the direction that is downhill. Right? So usually one direction is favored over the other. That's not always true, but usually one is favored over the other, and it's often in the downhill direction. Okay. So other ways to name enzymes, we've come up with, since we've discovered so many of them, we have a more rigorous naming system using numbers. This is akin to the way we were talking about our fatty acids. Remember, instead of naming them all these flowery names like arachidonic acid and stearic acid, we started naming them with a more numeric system that said, okay, it's got so many carbons and then so many carbon-carbon double bonds, and then tell me where those double bonds are. It's a really rigorous numeric naming system. And we've got similar thing for enzymes. It's done by the Enzyme Commission, which was developed in the 1950s, and they've updated it ever since. I think we're on the 7th edition now. where we've added a 7th category recently. But we're going to study the 6 that are well known. You don't have to worry about the 7th one at this point. And I will have you learn all 6 categories. Now, do you need to learn all the other minor contributors of those categories? No. I just want you to know the major 6 categories. So excluding the new 7th one that's been added in the last couple of years, I want you to learn the 6 major groupings of enzymes. And we'll talk about those in just a minute. So the way they're grouped together is by what kind of reaction they perform. Right? And we can break almost all enzymes down into 6 major categories. And that's generally given as the first number in this EC rating. Right? Our enzyme commission number. So it's kind of like the enzyme's social security number or phone number. Think of it that way. So the first number is the only one I really want you to know. It's either going to be 1 through 6. And I don't really care if you know which one's number 1 and which one's number 4 and which one's number 6. Just know that there are 6 of them I want you to learn. And so that first number tells me the type of reaction it's doing. So if it happens to be number 1, again, you don't have to memorize the number, but then it's an oxidoreductase. If it happens to be number 6, it's called a ligase, right? We're going to learn all 6 of them, but know that that first number tells you what major branch it's in. Every subsequent number, and there are 4 numbers, every gives you a little more detail or breaks it down a little closer to its unique reaction. So I gave 2 examples here, the 2 we were just talking about. Alcohol dehydrogenase. We said this is an enzyme, the ACE ending, that oxidizes alcohols, right? Remember, dehydrogenation, removing those hydrogens, makes a double bond, and that's going to be an oxidation reaction. So an alcohol dehydrogenase is an oxidoreductase reaction. It does redox chemistry. That's all this 1 stands for. But there are many enzymes that do that. So the second number 1 tells me, okay, it does redox reactions on primary or secondary alcohols. Right? That's very specific because not all reactions deal with those alcohols. So the second number gets a little more specific. Yeah, I don't care if you memorize this particular example. I just want to show you the differences in the numbers. The third number says, okay, of those that do that, of those that act on alcohols, do they use a specific cofactor? Remember, these are our copilots that are involved. And if it's a 1 here, it uses NAD or NADP as the cofactor. Other enzymes may have a 2 here. They use a different cofactor, or a 3 and a different cofactor. So it delineates those. And then the last number, the 4th number in these, is the individual enzyme from that particular organism. Right? So if you say it's 1111, which is the first one classified, it's an alcohol dehydrogenase, clearly that uses NAD or NADP as a cofactor to oxidize a hydroxy group. Right? And this particular one is called number 1. Number 2 would be very similar to it. It'd have all the same features, but it's a different enzyme. Right? Maybe from a different organism. Right? With a different sequence. So that's all I want you to know about these EC numbers. Is the first number tells me something broad category, what type of reaction it does. For example, down here, this one's a ligase because it's number 6. And then the next numbers get more specific. You don't have to remember the specifics at all, but I want you to know as you go down, it gets more specific. And the last number tells you this very individual enzyme, like it's its own social security number. Okay, so you don't have to memorize these 2 examples at all. I'm just using them to illustrate what these numbers mean, and that's what I want you to know. So if 2 enzymes have different numbers in the first position, they do completely different chemistry. But if they have the same number in the first position, they do the same type of chemistry. How they go about that chemistry is indicated by the other numbers, right? So if it says 1, 1, and then the next one says 1, 2, then it's doing an oxidoreductase reaction, but it's not on a primary or secondary alcohol. It's on something else. That's what these numbers represent. Okay, so what do I want you to know out of this? I don't want you to memorize these 2 examples at all. What I want you to know are these broad 6 categories, right? So these are the 6 I want you to know. I don't care if you associate them with their numbers or not. You don't have to know that transferase is 2 and isomerase is 5. I don't care about the numbers. I want you to know there are 6 categories and be able to tell them apart. So I wrote really short versions of their descriptions here on the right. So some of them are very simple, right? So an oxidoreductase, this should say an I here, but an oxidoreductase does redox reactions, oxidation reduction reactions. That should make sense from the name. That one's fairly easy. So it's usually a transfer of electrons. And I want to avoid using the word transfer here because you might start thinking about transferase. So how about an exchange of electrons? We say it that way instead, so I don't confuse you with this one. So an oxidoreductase is an enzyme that facilitates an exchange of electrons. right? So it does redox chemistry, right? And that's the vast majority of enzymes we're going to find. They're in this category, and that's why it was number 1. Okay? If it's not a redox chemistry, then it might do something else. So there's no redox involved for the rest of these, because if it does redox, it's in this category. So no redox in the remaining 5 categories. So if it does that, it's in number 1. It's kind of the top category. The others are also fairly simple. A transferase transfers groups, and I mean something bigger than a hydrogen. If it's transferring just a hydrogen, and it's probably transferring that hydrogen with electrons, and that's going to be back in our oxidoreductase. So transferases move things bigger than hydrogen. Right? It's not hard to find things bigger than hydrogen because everything else is bigger than hydrogen. Right? So that one's fairly simple. A hydrolase does what it sounds like. It breaks a bond, hydrolysis. Right? Lysis means to break, and it often does it using a water molecule. We've seen this before. Right? When we talked about breaking apart an ester, right? Or breaking apart a fatty acid, a fatty acid from the triacylglycerol. These were all hydrolysis reactions. We named a few enzymes back there when we talked about it, right? We had those when we talked about in the toxins, or when in the cell membrane that pulled apart our triacylglycerols or diacylglycerols. We talked about those type of enzymes. But when I say here it does hydrolysis, I mean lysis, and it often uses water, but it's not necessarily always water. Okay, keep that in mind. Just because you see water, don't associate it with hydrolase right away. That is not always true, right? And you're going to see lots of questions that'll challenge you on that. So if you see a water molecule involved, don't immediately think hydrolase, but it's a possibility. It can use other things besides water. We can use ammonia. We can use phosphates, and we'll talk about a few of those later in metabolism as well, right? But what we're doing with hydrolysis is breaking a bond. We have a single bond connecting 2 atoms, and we break it. So what was one molecule is now broken into 2 pieces. Right? That's what a hydrolase is. It breaks a single bond into no bond. Right? Fairly simple to remember. Doesn't always necessarily use water. Right? I'm going to skip lyase for a minute because it's probably the most difficult one to understand, and we'll move on to isomerase. And I'll come back to lyase at the end. I want to save it for last. So the isomerase is pretty simple. The name gives it away. It's an enzyme that turns one molecule into an isomer of itself. So there's no net gain or loss of atoms because isomers must have all the same parts. So all an isomerase does is convert one molecule to an isomer of itself. Right? Fairly easy to see when all the parts are still there. But of course it can't do any redox because that would be class number 1. Right? And if it did redox, it would have an alternating number of atoms, not what you started with, and that would not be an isomerase anyway. Okay? So that was fairly simple. Isomerases make isomers. Right? And then the last one at the bottom, ligase, makes new bonds. It ligates, it makes a ligature, right? That's to link things. So ligation is to put new things together. It's kind of the opposite of the hydrolase, where hydrolase was to tear things apart. So a ligase is to put things together. So it forms a new bond, like a new carbon-carbon bond, or carbon-oxygen, or carbon-nitrogen. These are single bonds. So just like the hydrolase, but in reverse. It turns a no bond into a single bond. So there was no bond between something, and now there's a single bond between them. The ligases often use an energy source for that, such as a molecule of ATP, right, or some other energetic molecule where it could break a bond there to facilitate making this new bond, but it doesn't have to. So just because you see ATP, don't associate that exclusively with ligase, because that's not always true. Right? All right, and then lastly, the one I skipped, let's go back to lyase, because lyase is kind of the none of the above category. It doesn't fit any of those others very well, but it's a choice of exclusion. If you can't make it fit any of the others, it might be a lyase. And there's only 2 requirements to be a lyase. You must not be an oxidoreductase, number 1, right? Because then you would be an oxidoreductase. So you can't do any redox reactions. That's true for all of these, right? So a lyase obviously cannot do a redox reaction, but it must make or break a double bond. So in other words, it turns a double bond somewhere in the molecule into a single bond, or it turns a single bond somewhere into a double bond. It must do that to be a lyase. Now, what the bonding atoms are is sort of irrelevant. It doesn't matter what those elements are, but it must form a single into a double or a double into a single without doing oxidation reduction. That's what a lyase is. So it's a very strict definition. It's usually the last choice of the choices of enzyme classification because it tries to fit the others first. If it does any of these other things, then it's that. Right? If it doesn't fit any of these other things, then we can call it a lyase. Right? And I'll just give you some examples as well. Right? Any questions on these 6 classifications? These are the short versions of their definitions. Okay.
Question real quick.
Uh-huh.
So is lyase the only one that deals with double bonds?
No, not necessarily. If you make a double bond and you did so by oxidizing something, right? So if you turn an alkane into an alkene, right? You removed a pair of hydrogens and you made a double bond. That would form a double bond from a single bond, but it's not a lyase because you did oxidation. That would be an oxidoreductase. Right? I have a little trick to help you understand what is a lyase versus an oxidoreductase, and I'll talk about that one later too. Right? It's a very simple counting scheme you can do to figure it out. Right, other questions? All right, so let's look at how enzymes are modeled. Right, so the first step of any enzymatic reaction, no matter what the enzyme might be, is it must bind to another molecule. It must bind to its substrate, we call it. Right, this is the reactant. It may have one or more, has at least one, but it has to bind to it. There's no action at a distance. It can't send a text message to its substrate and say turn into product. It doesn't work that way. Right? It must actually physically make contact. So the first step of any enzymatic reaction is the enzyme must bind to the substrate. So we need to come up with some terms for this. When you have your enzyme, right, that is not bound to substrate, we call it the free enzyme because it's not bound to something. And of course the substrate is often referred to as just the substrate, or it could be called the free substrate because it's not bound to the enzyme. But just saying substrate is the same thing. So these two must bind one another. Right? That's the first thing that must happen, is the enzyme must bind to the substrate. And when it does bind to it, right, we call that the enzyme-substrate complex because they're bound together now. It's often abbreviated the ES complex. You're going to hear me say that a lot. Right? So the enzyme plus the substrate, the free enzyme plus the substrate, are often abbreviated E and S, form the ES complex. Right? Where the substrate binds, right, there's a very specific pocket here that the substrate binds to, we call that the active site, because that's where the action takes place. That's where the chemistry is going to take place. Of course it is. The chemistry takes place where the substrate is because it's acting on that molecule. Right? So that's all this active site means. It's the amino acids that line this pocket, right? Could be a shallow pocket, could be a deep pocket, could be hydrophobic, could be hydrophilic, could be any number of requirements. But it's the place where the substrate binds and the chemistry takes place. Right? So it's a three-dimensional spot on the enzyme where this action takes place. That's why it's called the active site. Okay? There's a lot of terms that use the words active site inappropriately, and we'll talk about those next time. Right? But where it binds is called the active site because that's where the action takes place. And so when it binds together, you get an ES complex. The chemistry happens, right? So in this example, we're cutting this molecule into 2 pieces perhaps, right? So whatever the chemistry here is probably a hydrolase as an example, because I'm cutting this molecule into 2 pieces. But it's doing the chemistry in this step, and now I have my enzyme product complex. So the substrate has become the product or products, if there's more than one. Right? But it's still bound to the enzyme, so it's still a complex. It's called the EP complex. P could be more than one thing, of course. So could substrate. But then you must release those products, right? So that the unbinding must occur, the release. So the products leave, and here they are, right? And it leaves you with a free enzyme again that's no worse for wear, and it looks just like the one you started. If it doesn't look like the one you started, it's not finished with its reaction yet. So if it doesn't appear to be exactly how it started, it's not acting as a true enzyme. It must get back to this state. And for most enzymes, as soon as the products leave, it is back in that state. Right? That's the ones I want you to be concerned with. So our enzyme binds the substrate first. First thing that must happen every time. It does the chemistry step, turning this into products, whatever that chemistry might be, one of our 6 types. right? And then it releases the product or products, making a free enzyme again. And this free enzyme is free to go back and bind another substrate. Does that make sense so far? That's all we're doing. Bind, chemistry, release. And then I can do it again and again and again. The enzyme is not damaged in that process. It's a catalyst. Okay, so there was two theories on how this happens. The first was by Emil Fischer in the end of the 19th century, and he called it the lock and key model. Right? Let me explain what he meant by that. He said for every substrate enzyme pair, right, because enzymes were specific for their substrates, they knew this. It wouldn't just use any molecule, and you guys told me that earlier today. So there's an active site that matches the binding of the substrate. In this little cartoon here, it shows the parts have an appropriate glove that they fit in here, right? So it matches the corresponding pieces. So he said these fit each other like a lock and a key, right? So there's a very specific key or substrate for every enzyme or lock, right? And that's how they find each other, right? It wouldn't fit any other lock, and this doesn't fit any other keys. So he called this the lock and key model of substrate binding. And it fit the criteria for almost every enzyme they came across. So it was assumed to be correct. And about half a century later, Daniel Koshland came up with the idea that that's not entirely correct, because we found enzymes by the middle of the 20th century here that don't fit this model, right? So he came up with one called induced fit. Here, the substrate doesn't exactly mirror the site of binding, right? It doesn't look like this at all. This looks like it fits perfectly. That was his lock and key. Here, it just doesn't fit perfectly. But when this approaches the enzyme and begins to bind, the enzyme changes shape and molds itself to fit that substrate, right? And that's how a lot of enzymes operate. And so he said it was the induced fit model. So the enzyme is induced to fit this key or fit this substrate. Well, not any molecule would do. It has to be the appropriate substrate to make it fit, because if it's just completely wrong, it won't fit at all. Right? So who was right? Is it the lock and key model or the induced fit model? Which one do you think is right?
Is it both?
Exactly. They're both correct. And that's true for a lot of theories in science, where one comes up with an idea that's a little limiting. It doesn't cover every case. It covers many cases that have been observed. And someone else may come up with a model that covers a lot of the other cases, but may not fit well with the first scenario. And oftentimes parts of both theories are correct, and that's what's true here. And in fact, many enzymes use a little bit of both, right? So some part of it might bind perfectly, and it molds itself to fit other parts of the substrate, right? And that's true for most enzymes. Many enzymes are purely lock and key. And that's the ones that they were studying at the end of the century here. And they fit exactly like this, and nothing molds itself to fit anything else. They're very specific. But some enzymes can use multiple different substrates, right? There's an enzyme called hexokinase, which is the one that was the poster child for this. And it's an enzyme that uses any 6-carbon sugar, that's why it's called hexokinase, as a substrate, not just glucose. So all the other sugars with 6 carbons in that arrangement will also fit. Some are better than others, of course, but they all fit. It can be used as substrates. Whereas something that's like a 7-carbon sugar or a 5-carbon sugar wouldn't fit. Right? So this one, he had to induce this, or come up with this induced fit model to explain why this enzyme is able to use multiple different substrates, because that's not allowed in this model. This was lock and key. You can't put the wrong key in the lock. Right? So this was why he came up with that idea, but it's not the only idea. This is also true for many enzymes. So it's a combination of both, as you said, that explains all the enzymes we've seen. So they're both correct in one way or another. And they were both wrong in saying that theirs is the only way to do it. Okay, but what both of them absolutely got right is the first step of any reaction is you have to bind your substrate. You have to make that ES complex. That's absolutely true. Okay. So let's talk about some factors that change the enzyme activity. So you can imagine you have a solution with your substrate in it, right? A water-soluble solution, because these things happen in solution, right? They're not happening in sand or in the air, right? So, and you have an enzyme there. It's a water-soluble enzyme. Most likely they're globular shaped because they're water-soluble. And so the enzyme's coming in contact with its substrate and then releasing the products and doing it over and over and over, right? And the factors in play could have an effect on that reaction. And one of the factors we're talking about is the temperature of that solution. And really, what does temperature really mean? Right? What's a really good definition for temperature? What does it mean is happening?
How fast the molecules are moving?
That's the best definition, exactly. So it's how fast things are moving, and if things are moving more quickly, what's the consequence of that? I can think of two consequences for moving quicker through solution.
They're bumping into each other more often.
Yes, they're contacting and bumping into each other more frequently. That's true. And one other thing. When they bump into each other, how is it different?
More energetic collisions.
Yes, they have more energy in those collisions. Absolutely. And so remember, having energy is a good thing. You can overcome that activation energy in that case. Okay? So really high temperatures, you don't even need the enzyme around. This reaction could take place. So going back to our sample of wood just sitting there on the floor, right? In this oxygen atmosphere we have, the wood doesn't spontaneously combust because you need a spark to get it over that first hump, right? Even in the absence of an enzyme. But if you raise the temperature in that room, right? So you put that piece of wood in a furnace, like no fire, just like an oven, right? Like say an electric oven, where it gets really hot, but there's no flame or spark or anything like that. The wood could spontaneously catch on fire because you've increased the temperature or the energy of the collision so much that you allow the reaction to occur, right? So temperature could have an effect because it allows the molecules to move around more quickly. And as far as an enzyme is concerned, how does that help? Let me go back to this picture here. How could moving molecules around more quickly facilitate an enzymatic reaction? What part is it helping?
They have a higher chance of bumping into each other.
They do have a higher probability of bumping into each other, but what else is also true besides bumping into each other? A higher temperature allows what else to occur? Look at this picture. You're telling me about this part, which is great. That can happen more often, I agree. What else could happen?
They'll bind together.
That's this part here, correct. They run into each other more frequently and bind, I agree. But temperature can also have an effect on the other end of this thing. What's happening down here?
They'll also come back apart.
Yeah, they have more inherent energy, right? Not just molecules flying around bumping into each other, but they're also vibrating more, right? They're jiggling around in here. And with more energy, because of a higher temperature, they're likely to fly out of here as well, right? So they can also be released more easily, as well as bind more frequently. So all those will facilitate this reaction happening more often. Whether an enzyme is present or absent, it's true. So even with an enzyme present, temperature has an effect. So for most enzyme catalyzed reactions, the higher the temperature, the faster it goes. And remember, by faster, what I mean by that, I mean it occurs more frequently. Right? But there's a limit to this. And if I increase the temperature, right, it goes faster and faster and faster, more frequently. But I get to a point where it won't work anymore. So if you could imagine a graph, I want you to generate this graph on your own. Draw an x and a y-axis. The y-axis will be the rate of reaction or the frequency of reaction. And on the x-axis, draw temperature. What type of curve should you draw in that space? What would it look like? As the temperature goes up, what should happen to the reaction frequency?
Go up.
It should go up, I agree. And then will it keep going up forever?
No,
Why not?
because the enzyme will denature.
Right. So for a regular chemical reaction, that would be true. It would just keep going up and up and up forever until the molecules themselves decompose. Right? But for an enzymatic reaction, you can only go so high until the temperature, the vibrations from all that energy, cause the enzyme itself to fall apart. Remember, it's held together by a lot of weak interactions, hydrogen bonding, ionic bonding, maybe a disulfide here and there, but it's not a permanently stable thing. It's not a chunk of iron, right? So it's oxygens and nitrogens and carbons and hydrogens mostly held together by these many, many weak interactions. So a high enough temperature and it falls apart. We call that denaturing. right? So when it falls apart, it kind of unfolds, and we said that's very unlikely to refold properly, and so the enzyme is becoming useless now. It fell apart. It's dead, right? If you want to think of it in that sense. So if you keep drawing your line, as the temperature goes up, you're going higher and higher on your y-axis, and then when you reach the point at which it denatures, what will happen to the activity?
It's like done.
It'll decrease pretty rapidly.
Right, it plummets to zero basically. So you should draw a hill that's continually going up, and then when it reaches the denaturation temperature where it unfolds and it can't do the reaction anymore, it should plummet all the way to zero. Right, the reaction will become inactive. It won't happen anymore. Right, so that's the temperature curve. It should gradually go up and then plummet to zero at some point. So at what point will the reaction be optimum, or the best place on that graph?
Like right before the temperature where it denatures.
Exactly, the highest temperature right before it denatures, right? So just a few degrees before denaturing would be the optimum temperature. Well, that's a little dangerous, isn't it, right? Because if the temperature goes up just a little bit, it won't work. So most enzymes have evolved to not operate at that temperature, but a little low below that temperature, right? And that's one of the reasons you get a fever, right? So your enzymes are operating ideally at something like 40 degrees, right? Or 41 degrees, or 42 degrees, right? Maybe even 45, a little higher. But if you go much higher than that, we're talking Celsius here. So if you go much higher than that, your enzymes start to fall apart. Right? So your body's at around 37 degrees, which is near the optimum, but not so close that it might cause them to fall apart. So you get very efficient enzyme function, rather than at, say, 20 degrees, which is not very efficient at all. So if you're infected by another organism, like a bacterium, right, or a fungus, or a parasite, or something like that, and its enzymes are also functioning, your body attempts to raise the temperature temporarily, not permanently, to say 42 degrees. Right? And that's dangerous for your enzymes because you're going to start to denature some of your own. But you're also going to denature all those bacterial enzymes, right? Because they're not so easily tolerant of that high temperature of 42 degrees, where it's normally 37. So if you increase it that high, just temporarily, preferably not in the brain, but other places, it temporarily goes up, you can help your immune system clear the infection. Your enzymes, maybe some of your cells are dying because of this as well, but we'll replace them, assuming it's not nerve cells. But the bacteria can't replace theirs fast enough, and your immune system is fighting it and wipes out the infection. So at some point, your body realizes that I've kept up this fever for a while. Hopefully it's been long enough to help the immune system out, and now we need to bring the temperature down before we cause permanent damage to ourselves. That's your fever breaking. And your body resets its thermostat back to 37 degrees, and you start sweating profusely to bring your body temperature back down. That's what we call the breaking of the fever, right? It doesn't mean you're over the infection. It means your body has tolerated this temperature long enough, and if it hasn't cured the infection by now, we're going to have to do it some other way. We can't keep having this high temperature. It's dangerous for the body's own cells, right? So that's why you have a fever. It tries to denature the enzymes of the invaders more so than yours. Another example of that is when we sterilize instruments in the lab or tools for surgery in something called an autoclave. Right? It's a big chamber full of superheated steam. That's all it is. It's a very simple machine, in fact. It's got a lot of fancy electronics for timing and things, but it's just a big chamber of superheated steam, and it holds it at high temperature and high pressure for, say, 20, 30, 40 minutes. And anything in there will be denatured. That's the idea. So if there's any infectious agent or viral particle, bacteria, parasite, whatever it might be, there are very few hardy viruses and fungal spores that can survive it, but not very many at all. So you sterilize the tools this way to prevent infection, because you're going inside of someone's body with these tools, or you're using them for something in the lab that needs to have no other bacteria around, because you can contaminate your results. So we use autoclaves for sterilization from high temperature and high pressure to make all these enzymes and other proteins fall apart. Okay? Some organisms have decided that they want to live in these high temperature environments. These are called thermophilic organisms, or thermophiles, right? And they live in very high temperature environments, such as if you go to Yellowstone National Park and you look at the geysers that are there, that water is near boiling, right? So there's organisms that are living there in these 60, 70 degree Celsius water pools, which would kill us, denature our enzymes instantly. but they're living there. They're colored. You have all these bacterial colonies that are living there. And so they must have enzymes that are stable at those temperatures, right? And so that's why we've discovered these thermophilic organisms, because if they're living in this temperature and thriving, then their enzymes must be stable at those temperatures, right? There's more interactions holding their enzymes together, whereas they don't grow so well at lower temperatures because they've evolved to live in higher temperature environments. Right? Besides temperature, pH can have an effect on enzymes. And this could make sense because if you change the pH, change the number of protons in the environment around an enzyme, it may need certain amino acids, and you're still learning your amino acids, I hope. Some of them can act as acids, some of them can act as bases, and they get different protonation states we talked about, right? At different pHs, they have an extra proton or they've lost their proton in the cases of the 5 acids and bases we talked about. But it may need to be protonated or may need to be deprotonated for the enzyme to function, right? So imagine if it's a carboxylic acid group like an aspartic acid, right? That's COOH on there. It may need to have that H removed, the proton removed, and become a carboxylate with that negative charge for this enzyme to function. So if you make the environment too acidic, it'll put that H back on, and then the enzyme won't function. Similarly, you could have the same argument for the basic amino acids, like a lysine. Right? It's normally an NH3 plus at the end of that carbon chain of the R group of lysine. But if you make it too basic, it'll lose that proton and just become NH2. And without that positive charge, it may not function as an enzyme. And the reverse could be true for enzymes as well. But so if you look at a graph again, I want you to make another imaginary graph. Y-axis again is activity of the enzyme, or the function of the enzyme. And on the x-axis this time will be pH. So could you imagine, much like temperature, there's an optimum pH at which this enzyme works best, right? And if I deviate from that optimum in either direction, more acidic or more basic, it's not going to function as well, right? So if I go too far, I go to extremes of pH, then the same thing's going to happen as if it were to temperature. If I make it far too acidic or far too basic, the enzyme is going to start falling apart, because remember, it's still held together by all those weak interactions, like hydrogen bonding, in particular ionic bonding, which requires those charges. If I change the pH, those charges disappear or become different, and it won't hold together as well. So if you want to draw your graph for pH versus enzyme function or enzyme activity, you pick a high point somewhere on the graph. It's not necessarily 7. It could be anywhere on the graph. And if I deviate from it, it comes down, so it's making a bell-shaped curve, right? And if I go too far, it plummets to 0 again. Right, so there's an optimum pH, just like there was an optimum temperature right before it denatured, but the optimum pH is far from the denaturation point, not next to it. Right? You have to get to extremes of pH to cause it to denature. Right? And this is the case for a lot of our digestive enzymes like trypsin, pepsin, chymotrypsin, those older enzymes that have been studied. Right? You're going to find pepsin in the stomach. It functions very well at pH 1 to 2. But as it goes through the stomach and digests your food there, ultimately making its way into the duodenum and the small intestine, the pH there plummets to something like pH 7, pH 8 even. Right? So it's not going to function very well at that pH, and in fact the trypsin, or the pepsin itself, is digested as more food. Right? It served its purpose, we're going to regenerate, we're going to recycle it. So enzymes are functioning in the environments from which they have evolved to, and if you deviate from it, they don't function well. So 7 is not always the optimal. Don't let 7 fool you. It's no special place on the pH scale. Right? It doesn't mean neutral at all. Right? A pH can be very acidic. 8 can be very acidic compared to 12. It's always a comparison. So don't think any one number
I have a question.
got a favored position. Yeah, go ahead.
When we talk about optimum pH, is it the same optimum pH for all enzymes, or does each enzyme have a different optimum pH?
The latter. Each enzyme will have a pH at which it operates the best. So there's no one universal optimum pH.
Okay, thank you.
Right? Just like there's no optimum temperature for every enzyme. Everyone's quite different. But each has an optimum, is what I'm trying to say. Right? An example of that is shown below, and I think you're going to do this later in lab. I'm not sure because we're not on campus this semester. I don't know if she still has the plates I made for her. But if you try to make Jell-O, right? So you've probably made Jell-O before. It's a gelatin dessert, right? It's a a molecule that you heat up, right? It's got gelatin in it, and you put in all your other ingredients like your sugars and fruits or whatever you're going to put in it to give it flavor, and then you let it cool down. And as it cools down, this forms a semi-solid, right? That semi-solid is made of a gelatin molecule which has bonds holding it together, right? And these bonds look a lot like the peptide bonds of proteins. So if you add a digestive enzyme like pepsin or trypsin or any of these other proteases, we call them, they will digest the bonds of the gelatin, and it will fall apart. It'll never solidify. As it's trying to form the semi-solid, the bonds get broken by these enzymes and it stays a liquid. Right? So if you look on the back of a package of Jell-O, it specifically tells you, do not use fresh fruits like pineapples, kiwis, papayas, figs, because they have a high concentration of these proteases. Right? And you've done this, if you want, you can test it for yourself. Get a canned pineapple, right? Buy some canned pineapple, or a kiwi or papaya or whatever, any of these others that have high protease concentration. Bananas would not work very well, and apples don't work very well. But these pineapple's very good at it, or kiwis. Buy a fresh pineapple and canned pineapple, and do this experiment. Take a piece of the canned pineapple and hold it in your cheek for a while, right? Don't chew it up or anything. I mean, chew it a little bit, but then hold it in your cheek, right? Like break open the fruit and hold it in your cheek for a while. And then feel how your cheek feels after you've done, after a minute or two. And then do that same experiment, obviously not immediately afterwards or in the same cheek, use your other cheek, for the fresh pineapple, right? And you'll notice in the fresh pineapple, your cheek feels a little worn, right? As if some damage has occurred. And it is. The pineapple's enzymes have tried to digest some of your proteins in your cheek. That's what gives it that rough texture after you eat those. It's not completely harmful for you unless you leave it in there for hours, but you generally don't chew something for that long. But it's also why you can't make Jell-O with them, because those fresh fruits will also digest the gelatin molecules, and it'll never solidify. Why will canned work? What's the difference?
I guess because it's fresh, it's higher acidity. So when it's frozen, it's become cold, so the pH changes.
Okay, that's usually the answer I get. It's not entirely true, because the pH does not change whether you heat something up or cool it down or put it in a can. And you're going to see that. That's the very experiment you're going to do in lab, that the pH and the acidity doesn't change. What's changing here is those enzymes, those digestive enzymes that are found in the fresh fruits. When they can these things, they usually do heat pasteurization. They heat them up really hot, and the idea is not to destroy these enzymes, it's to kill off any bacteria that are in it before you put it in the can, because you're going to leave it in the can for storage for a long time. Right? You can't store fresh fruit forever, but you can store a canned fruit for quite a long time if you kill off any of the bacteria. So they use heat pasteurization. They heat it up really hot, you boil it or whatever for several minutes to 30 minutes to an hour or something like that. If you've ever seen your grandmother make canned jellies, you'll understand. And then you let it cool down and you put the can's lid on, right? And because of that, there's no bacteria and it lasts a long time in that can, right? But because you've heated it up to that boiling temperature and cooled it back down, all those digestive enzymes in the fruits have been denatured. So when you put it in the Jell-O or you put it in your cheek, the acidity is still there. It's very acid still, right? And you'll see that on your plates when you do that in lab. The acidity is still there. It can still denature proteins, but it's not digesting them, so it doesn't fall apart. And that's the difference. So if you have a catering company later in your life and you have the bridezilla who's coming up and asking you, I must have kiwi Jell-O at my wedding, buy canned, not fresh, and you won't have the Jell-O that will never set. Right? But what if you only have fresh pineapple available? Is there a way to use fresh pineapples in your Jell-O?
You'd have to cook it.
Cook it first, right? Boil that pineapple for a couple of hours first, right? It won't change the taste of the pineapple much. It won't change the acidity, but it will denature those enzymes so that you can make your Jell-O out of it. Good. All right, some other factors that influence enzyme activity here is substrate concentration. So we did temperature, we did pH. The amount of substrate available can affect how the enzyme works. And you can imagine that if there's very little substrate available, right, low concentrations, not a lot of it in the solution, the enzyme is going to encounter it rather infrequently, right? It's not going to run into it very often. So you're not going to get a reaction very often. If there's lots more substrate around, it's going to encounter it more often, and you'll get a more frequent reaction. That makes common sense, right? And in a simple chemical reaction, that gradient, or the increase in rate based on the concentration, shown here on the left, is a straight line forever, right? So the more reactant you throw in the solution, the faster it will convert to product, right? A larger barrel of fuel, bigger explosion, right? So that's a simple linear relationship. That is not the case for an enzyme catalyzed reaction. This is not a straight line. You see it kind of curves over, right? Has this hyperbolic shape, not a straight line, not linear. It doesn't go up like this. Why? Can you give me a reason why, if I add more substrate, the reaction will always get faster. You notice this line never dips. It always gets a little faster no matter how much substrate I add forever. It always gets a little faster, but it's got some diminishing returns. It doesn't get much faster. And it seems like eventually it's going to fall over and make a straight, flat line. And it will. What's going on?
Is there like a limit to how fast an enzyme can bind and release the substrate?
That's exactly right. So in order for a simple chemical reaction to happen, you just have your substrates turn into product, right? By automatically doing it. There's no enzyme involved. But in an enzyme catalyzed reaction, what must happen? The substrate has to bind to the enzyme, perform the chemistry, and then let the products go. No matter how much substrate you throw at it, if you throw an infinite amount of substrate at that enzyme, it still has to do that, and that takes time. So because you're relying on another molecule that is not infinite in concentration, the enzyme itself, this is for a fixed amount of enzyme, it can only go so fast. Right? And I'll give you an analogy for that. If all of you were sitting in a classroom, right, and there were tons of sheets of paper on the desks in there, and I gave you each a pair of scissors, and I said, I need you to cut out a little figure, right, like a whatever shape you'd want to do, a star, a circle, a gingerbread man, whatever you'd like to do, cut out a shape from each of those pieces of paper. Right? I gave you all scissors to do it. And the more paper I throw at you, the faster you could do it probably, because you're not looking around for the next sheet of paper. But there comes a point where no matter how much paper I throw at you, you can only go so fast, because you're required to pick up the paper, cut it, release your product, and then grab another piece of paper. Right? So that's why an enzyme can only go so fast. Can I make that reaction go faster? Sure, add more students, right? More people with scissors could make it go faster. But that's not a fixed amount of enzyme. That's adding more enzyme, right? So for a fixed amount of enzyme, it can only go so fast. Does that make sense on this graph? So it's the rate of reaction for a fixed amount of enzyme with more substrate can only approach a maximum. Okay? The opposite is true. I'm going to skip this slide for a second and go back. The opposite is true if I add more enzyme. Right? This is more students with scissors. Right? The more paper I throw at, for a fixed amount of paper, right? The more students I add to the room, the faster we can make cutouts. Right? And that's true no matter how far I go. Right? If there's an infinite amount of paper in the room, as long as I keep adding students and you fit in the room, we can keep making cutouts faster. Right? So the rate of the reaction is proportional to the amount of enzyme, but it is not proportional to the amount of substrate. There is a maximum we can achieve. Right? You can't go any faster than that because it requires you to bind, do the chemistry, and release. Does that make sense between these two? Be careful when you're looking at graphs. I want you to read the axes. This is the rate of reaction as I increase the amount of enzyme. That's going to be linear forever. More students with scissors. Here, I have a fixed number of students with scissors, fixed number of enzyme molecules, and if I add more substrate, more paper, you can only go so fast. I can throw tons of paper at you, but you can only move so quickly, right? Because you have to bind, cut, and release. Those two graphs make sense to you? Right, so this is the maximum rate it can achieve, right? And we're going to look at some maximum rates for several enzymes. These are called the turnover numbers. How fast can one enzyme do this? Right, so given an infinite amount of substrate around, right? We have substrate molecules that will never run out, right? Think of it as an infinite concentration. How fast, this is a theoretical calculation, of course, but how fast could the enzyme go? How many products can it make per unit time per enzyme? And some enzymes are faster than others. right? So carbonic anhydrase, which is an enzyme that converts carbonic acid into CO2 and water. It happens in your blood to convert the dissolved carbonic acid into CO2 so you can exhale it in the lungs. Has to do that fairly quickly so you can get rid of the CO2 in the brief window that it's allowed to in the lungs. So it does 600,000 of these per second per enzyme. It's incredibly fast. This is 600,000 enzymatic reactions of bind, chemistry, and release per second per enzyme. One enzyme does that many. Right? So there's lots of these carbonic anhydrases in the cells that are in your lungs as the blood passes by, right? And it turns it into CO2 so you can exhale it. Right? Some enzymes are not so fast, like lysozyme. It's very slow in comparison. It does one every 2 seconds, right? So half of one per second. So this is an extremely slow enzyme in comparison. By slow, again, I mean infrequent. It doesn't do it very often. Right? Of course, if you add more lysozyme, it goes faster, but for a fixed amount, these are their speeds per enzyme molecule. And this is what it evolved to do. Don't think lesser of lysozyme because it's not as fast as carbonic anhydrase. It evolved to do what it needs to do. Right? So this needs to go fast because it needs to convert all that carbonic acid into CO2 so you can exhale it quickly. Lysozyme punches holes in bacterial membranes. How many holes do you need to put in a submarine to sink it? I don't need to put 600,000 holes. One will do. Right? So this punches holes in bacteria. And as long as it can punch the holes faster than the bacteria can repair them, which it can't repair them very quickly, one every 2 seconds is quite fast for that, it's functional. It's sufficient. So they have evolved to the number they need for their function. Not necessarily, don't think lesser of this enzyme compared to this one. Okay, so these are just some examples of extremes of those. Right? And then the last slide I want to go over today is a little bit of the mathematics of graphing these parameters, and then we'll pick up here with inhibition next time. So let's go over these graphs real quick. And I posted a paper that was published a little over a decade ago on this exact thing and the inhibition that goes along with it. So I want you to read that before next time. I posted that paper for you on your list. So here we're going to plot a couple things. And you've seen this plot before. I just haven't called it this. This is the exact same plot we've been showing here, right? Where substrate on this axis and velocity or rate on this axis. Same one I showed here. Substrate concentration, velocity or reaction rate on this axis. And I'll show it one more time. Substrate concentration, S brackets mean concentration. So this is the substrate concentration and the velocity, V, or the enzymatic activity, or the rate of the reaction, right? Or frequency of the reaction. Whatever words you want to put there, that's just what this means, how rapidly it's happening. Right? It's the same curve, right? It's a hyperbolic curve. Right? It doesn't go straight, it's not a straight line, like a chemical reaction would be. It's this curved line. And if we said eventually it will curve over and get slower and slower, not slower, but it will get a less of an increase with each additional amount of substrate. Eventually this line approaches an asymptote, which is called Vmax. Right? So it's the maximum rate that this enzyme could achieve given an infinite amount of substrates. If you go all the way to the right on this graph, all the way to infinity, then this line will finally reach this point, but it'll never go above it. Right? So this is called the maximal velocity, or Vmax. So what I want to get across here is not memorizing something, because if you start trying to memorize something here, you will always get this question wrong. And I ask the question the same way on every test for the past decade. And without fail, about a third of the class always gets it wrong. Right? And I tell you not to pick it. It's going to be choice A. Don't pick choice A. Right? And it asks you to describe what is Km. What does this Km, or Michaelis constant, named after one of the authors, Michaelis constant, mean? Right? It does not mean it's equal to 1/2 Vmax. This is choice A on the test, and it is wrong. But a third of you will still pick it because you're just memorizing word association. Don't do that. What does this term really mean? So let me explain it to you. If this is the maximal velocity that it can achieve, right, I can calculate that by adding different amounts of substrate and measuring the velocity and putting some points on this graph. And then I see it's forming a curve and mathematically calculate what Vmax is. And then if I know what Vmax is, I can divide it by 2 and get what 1/2 maximal velocity is. Because that's not maximal, there is a certain concentration, right, at which I achieve that half maximal velocity. It's the amount of substrate. You notice Km is on the x-axis here, right? It's a substrate concentration. and it's the amount of substrate it takes to achieve half of the speed that an infinite amount would give me. Okay? And one of the ways to think about that, notice this is not a straight line, right? Keep that in mind. If I double the Km, right, double the concentration that achieves half the maximal velocity, I will not get maximal velocity, right? And that's because this is not a straight line. If you double the Km, so here's the Km, the amount of substrate it takes to achieve this velocity. If I double the Km, that would be here, right? So here's Km, here's would be double Km, about right here. And if I go up, that is not Vmax, that's this point right here, right? It's just a little faster, right? And no matter how much more multiple of Km I add, I'll never achieve Vmax until I get to infinity. That's the definition of Vmax. Because this is not a straight line. Don't think of it as a linear relationship. Don't make linear relationships out of it. But what can this Km tell me? This is more important. Km tells me the amount of substrate it takes to achieve a certain speed. Right? It happens to be the speed that's half of what you could do with an infinite amount. But if Km is a large number, relatively speaking, right, it takes a large amount of substrate. Just think about this in your head. If it takes a large amount of substrate to achieve this speed, then this is not very good. I have to throw a lot of substrate at it and make it go this fast. Right? But if Km were a very small number, right, say it was down here instead, then it would not take very much to achieve a large speed. Right? Oops, sorry. So the Km represents the amount of substrate it takes to achieve a certain speed. And if it were very, very small number, say down here instead, this graph would look different. It would go up to this point, because if Km were here, then it would go through this point right there. So this curve would go up rather quickly and then curve over, right? And that means it takes less substrate to achieve the same speed. How is that possible? How can you throw less substrate at something and get it to go just as fast? What must be true? So if I have two enzymes and I use the same substrate, one has a large Km, one has a small Km. Which one will go faster for a given amount of substrate?
The one with the small Km is more efficient.
It's not necessarily more efficient, but it does what? It does go faster for a fixed amount of substrate, which means what about that enzyme and substrate pair?
It binds and releases more quickly than the other one.
Well, it binds it more quickly. We don't know so much about the release, but it definitely binds it tighter. So because there's less substrate around, right, lower concentration, it encounters it less often, but it's going just as fast. How is that possible? Well, it must not let it go very often. Right? So it binds the substrate, and not always when you bind it, you turn it into product. Oftentimes you just let it go. Like a catch and release program with fish, right? So you don't always cook the fish. Sometimes you let them go. So enzymes are the same way. You catch the substrate, and it doesn't always turn it into product. It sometimes lets it go. And when it lets it go, all right, more frequently, then it doesn't bind very tightly. If it does not let it go very often, then it binds really tightly. So what does Km tell you about the enzyme substrate pair?
How tightly they bind to each other.
Right, and a term for that is affinity. Right? So if something has a very large Km, it has a very small or low affinity. It's a reciprocal relationship. Remember, the smaller the Km, the tighter it's binding. The easier it is to achieve that velocity. So the Km and affinity have reciprocal relationships. So a very large Km means a very weak binding, and a very small Km means a very tight binding for that enzyme substrate pair. That's what Km means. It doesn't necessarily equal 1/2 Vmax. That's not true at all. They're not even the same units. This is substrate concentration and this is velocity. That means nothing. Right? An absurd way to think about that is, how many spoonfuls of sugar do you need to eat to run 20 miles an hour? Well, the number of spoonfuls is not 4 miles an hour. They're not even in the same units. Right? So it doesn't make any sense to call Km equal to 1/2 Vmax. They're related in a way, but they're not equal to each other. Km is the amount of substrate it takes to achieve a velocity of 1/2 maximal. That's the correct association. So if you see it written any other way, it's not correct. Okay? And that's the same graph shown on the bottom here with the curvature and so forth. But this is a curved line. It's not a very good way to think about things with curved lines. And a pair of researchers named Lineweaver and Burke came up with a way of converting a curved line. This is a hyperbolic curve. We're not going to go into the mathematics for this course. But if you take the reciprocal of all the data, you end up with a straight line. Right? So this is a hyperbolic curve, and the reciprocal of a hyperbolic curve is a linear straight line. So if I plot it this way, I get a straight line. The axes are inverted, so instead of velocity, it's 1 over the velocity. Instead of s, it's 1 over s. But I get a nice straight line. And now some points stand out on the line. So, and this is where I'll leave you for next time, is when you have a straight line, it can be described by 2 numbers. What are the only 2 numbers I need to describe a straight line? Any line. If I give you these 2 parameters, you can draw it.
The y-intercept and the slope?
Exactly. So I need the y-intercept, which is right here, and the slope, which is the rise over run of this line. So given those 2 numbers, here's the y-intercept, here's the slope, the rise over the run, I can get the parameters for an enzymatic reaction. I can get Vmax because it's 1 over the intercept, and I can get Km because I can take the slope and the intercept and divide the 2 and get the Km. Right, so this divided by this will give you the Km. So I can get those 2 factors by converting all my data into a straight line. That's the Lineweaver-Burke plot. And from there I can calculate the 2 parameters of the kinetic equation, the Vmax and the Km over Vmax. Right, and that's also shown down here. Right, so we'll talk about that more next time when we get into enzyme inhibition.